Hello class, my name is Josh Dougherty and tonight I will be talking about the Salmonella. I will be discussing about the scientific name, the first scientist that discovered it, the taxonomy, the structure of the Salmonella bacteria, the pathogenesis, the epidemiology, and the treatments and preventions against Salmonella. The scientific name for Salmonella is also called Salmonella. In 1879, Carl Joseph Eberth, a physician and student at Rudolf Virchow, discovered Salmonella in the abdominal lymph nodes and the spleen. When he published his discovery in 1880 and 1881, German and English bacteriologists such as Robert Koch confirmed it. It was Daniel Elmer Salmon, a veterinarian pathologist, who gave the genus name Salmonella. Both Salmon and Theobald Smith found Salmonella in hogs that succumbed to hog cholera. So let's talk about the taxonomy of Salmonella. It belongs to the kingdom Bacteria, further categorized into the subkingdom Negibacteria. The bacteria is within the phylum Proteobacteria, to the class Gamma Proteobacteria, then the order Enterobacteriales, and then to the family Enterobacteriaceae. Many taxonomists consider Salmonella to belong to only two species. One species is the Salmonella bongori with one subspecies V. The other species is Salmonella enterica, which has 7 subspecies, 1, 2, 3A, 3B, 4, 6, and 7. Subspecies 1 infects warm-blooded animals, while the rest of the subspecies infects cold-blooded hosts. The Salmonella species is a gram-negative, flagellated anaerobic bacilli characterized by O, H, and V antigens. There are over 1,800 known serovars that are considered separate species. The Salmonella has flagellum formed by the FLIE component. The component is the first protein secreted through the flagellar type 3 secretion system that has the ability to self-assemble. The FLIE protein has 2 roles in assembling the Salmonella flagellum. The flagellum is the final component of the flagellar type 3 secretion system, and as an adaptive protein that anchors the rod of the flagellar motor to the membrane-embedded MS ring structure. When the Salmonella enters the host, it uses the flagellum for both motility and pathogenesis. The flagellar organelle is extracellular, and the type 3 secretion system secretes structural subunits which are assembled into the growing structure. Salmonella enterica possesses 2 other T3SSs, which are involved in secreting virulence-associated effector proteins. These effector proteins are secreted through injectosomes, which are protein hypodermic needle-like protein injecting structures. The proteins are sent into host cells to facilitate Salmonella infection. The bacterial flagellum utilizes more than 30 gene products for construction itself. The flagellar protein genes names are based on the chromosomal locations where they are located, the FLG, FLH, FLI, and FLJ regions. The flagellum has 3 main structural components: the basal body, which serves as the ion-powered rotary motor to anchor the structure, the hook, and the filament. Another interesting structural element of the Salmonella is the formation of strings, specifically in the subspecies Salmonella typhimurium. These 1 cm long string-like structures are induced under some stress conditions such as iron deprivation by 2,2-bipyridyl or low antibiotic. But cells and strings revert back to planktonic growth when they return to nutrient-rich media. Unlike the planktonic cells, which are susceptible to antibiotics, strings are more resilient to antibiotics and oxidative stress. Also, unlike strains with the CSGD or RPOS, which are defective in the classical RDAR biofilm formation, strains without a CSGD or RPOS form strings. The first step the bacteria make to infect the human body is to invade the intestinal mucosa and multiply there. In rare cases, Salmonella passes through the intestinal mucosa at M cells. thus entering the lymphatic and cardiovascular systems. Such an infection may spread and affect many organs throughout the human body. Salmonella multiplies in the body by replicating the macrophages. Salmonellosis develops within the body for about 12 to 36 hours. The disease leads to many symptoms such as moderate fever accompanied by nausea, abdominal pain and cramps, and diarrhea. But Salmonella encounters many obstacles after entering the host. Salmonella encounters many barriers such as low gastric pH, intestinal mucosa, and intestinal epithelial cell barriers. Salmonella can survive those barriers and cause infection efficiently. This is because of Salmonella's complex membrane structure, which enhances the bacteria's survivability until reaching the lower intestine's epithelial cell wall. Salmonella enters into those host cells through the microfold M cells of Peyer's patch. These M cells are masses of lymphoid tissue found throughout the small intestine that analyze and respond to pathogenic microbes in the intestine. Salmonella has two major ways to enter into the host cell. The first way is through the M cells by transcytosis, a passive process by which the bacteria are taken out by these cells from the lumen to the basolateral side. The second way is the uptake induced by the bacteria by secreting SPI-1 TTSS effector proteins, causing membrane ruffling by the cytoskeleton rearrangement of the epithelial cells. Additionally, antigen-presenting dendritic cells, or DCs, send processes out to the lumen through the epithelial cells without disturbing the cell junctions. These DCs help sample bacteria to nearby mesenteric lymph node. Invasion is mediated by the flagellar motility, along with the TTSS1 effector proteins. Various SPI1 proteins act together in facilitating the invasion process by needle complex assembly, effector protein translocation through this needle apparatus, and actin cytoskeleton alteration. These elements prepare evasion of Salmonella. The epidemiology of Salmonella explains why 1/3 of the infections are transmitted through food. In fact, many Salmonella cases, 52% of non-typhoidal and 30% of typhoidal Salmonella cases, compared to other enteric pathogens, are considered to be foodborne. Since many of them are foodborne, they can be prevented once vehicles are identified. Salmonella's epidemiology evolved over decades because people produced and consumed a greater amount of food as industrialization and globalization of food supply increased. Growing demands for ready-to-eat and raw or lightly cooked foods, aging populations, and higher proportion of immunologically compromised individuals also play a role in Salmonella's epidemiology. Tools for analyzing trends such as PulseNet have improved to analyze foodborne diseases and detecting outbreaks. PulseNet detects disease clusters that may represent Salmonella infection outbreaks. PulseNet aids outbreak investigations that by analyzing a wide variety of food commodities, such as various fruits, fine vegetables, leafy green vegetables, spices, tree nuts, poultry, beef, and much more. Laboratory-based surveillances can detect other types of trends, such as increased infections by reptiles and amphibian-related Salmonella serotypes. Such surveillances can even detect types that reflect increased adoption of these animals as pets. And of course, there are increasing quality and comparability of exposure data impacts that affect the outcome of analytical studies in a manner similar to increased resolution. If one examines a typical 2x2 table from a case control study, the WGS and other agent subtyping methods will help refine the case definition, thereby eliminating those cases less likely to share an epidemiological association. Scientists used outbreak data from a previous study used to determine the relative contributions of 17 different food commodities to the annual prevalence of foodborne illness in the United States. It was hypothesized that the exposure pathways of foodborne outbreak illnesses were representative of those pathways for all foodborne illnesses. But such assumptions cannot be tested directly because food sources of sporadic illnesses are unknowable. In fact, even though multiple cases and controls are available to examine the likelihood of illness for different foods consumed, such food sources are identified in only about 1/2 of all foodborne disease outbreaks. Data submitted to the Centers for Disease Control and Prevention by public health personnel from each FoodNet site indicate whether a case of foodborne illness is an outbreak or not. Scientists aim to determine whether differences exist in terms of 6 selected characteristics of outbreak cases of laboratory-confirmed Campylobacter E. coli 0157, Listeria, and Salmonella infection reported in FoodNet during 2004 to 2011. The 6 characteristics examined were as follows: the FoodNet site reporting the case, the year in which a case occurred, the season in which a case occurred, the age of patient, generally the difference between submission date and reported date of birth, the sex of the patient, and the hospitalization status of the patient, i.e. whether the patient was hospitalized within 7 days of the specimen collected. Since 2004, the FoodNet surveillance catchment area has been stable. The FoodNet sites were Connecticut, Georgia, Maryland, Minnesota, New Mexico, Oregon, Tennessee, and selected counties in California, Colorado, and New York. To ensure sufficient data, scientists determined quintiles for season and age groups. Because the data distributions differed between the pathogens, these quintiles were determined for each pathogen separately. Sex and hospitalization status were binary variables. To complete the analysis of these characteristics, scientists used a 2-step approach for each of the 4 pathogens examined. The first is that scientists conducted random forest and boosted tree analysis to gauge the relative importance of the 6 characteristics in distinguishing between outbreak and sporadic cases. Random forest analysis is a data classification algorithm that seeks the best combination of factors to explain an outcome variable. Boosted tree analysis pertains to the use of regression techniques. For measuring the fit of the trees to the data, scientists created random collections of classification trees and averaged those trees by a measure of how well each tree fit the data. The second step of the analysis was logistic regression modeling. Scientists used stepwise model building routines to examine all main effects and interactions among the factor levels. This explains the fraction of cases that are outbreak-associated cases. Scientists use forward selection procedures and minimum Bayesian information criterion scoring, BIC, as a model identification guide. This was preferred because it penalizes the inclusion of additional parameters more strongly than alternative statistics. Scientists then selected the logistic regression models with the lowest BIC scores as the best models. Seasonal quintiles were similar across pathogens except for E. coli 0157. The first season was longer compared with the other pathogens, extending from January through the end of May. But age quintiles differed substantially across pathogens. For example, to capture 20% of the data for Listeria, the first quintile was defined as class cases in patients who were 0 to 38 years of age. In contrast, the first quintile for Salmonella only extended to patients 3 years of age. For Listeria, the relatively narrow quintile range for persons 60 to 80 years of age reflects a larger number of older persons among the cases. A descriptive treatment of the data shows that the frequency of outbreak cases among all FoodNet cases varied more for FoodNet site, year, patient age, and season than for sex and hospitalization status for each pathogen. Compared with the other pathogens, Listeria exhibited substantial frequency ranges for some characteristics. In the scientist's analysis, the probability of a case being outbreak-associated varied significantly across the FoodNet surveillance sites for all 4 pathogens studied. Uncertainty exists for the causes of variability of the number of ascertained cases across FoodNet sites and the number of outbreaks detected and reported across states. The study concluded that the characteristics of outbreak and sporadic cases captured by FoodNet vary for all 4 pathogens examined. But more importantly, their finding with respect to age differences for Salmonella outbreak and sporadic case patients suggests that applying outbreak-based source attribution estimates to the youngest case patients might be inappropriate. Otherwise, since the analysis finds that outbreak and sporadic illnesses have similar case characteristics, the impression is that this study does not refute the plausibility of outbreak-based source attribution methods. Salmonella can be treated with a few home remedies. The main way people can recover from salmonella is if they rest and drink enough water. In fact, it is highly recommended to stay hydrated as much as possible. This is because Salmonella possibly causes vomiting and diarrhea. An adult should drink water, broth, sport drinks, unsweetened fruit juices, and any beverage without caffeine. However, severe nausea can hinder a person from drinking beverages. It's recommended to suck on ice chips to keep one hydrated. Dehydration occurs when one loses more fluid than consumed. When adults suffer dehydration, the following signs occur: excessive thirst, decreased urinary frequency or output, darker than normal urine color, dizziness, episodes of confusion, and extreme fatigue. While the patient recovers with Salmonella, certain foods should be avoided as they can worsen Salmonella symptoms. The following should be avoided while infected: alcoholic drinks, caffeinated beverages, dairy products, fried or greasy foods, sugary drinks, fruits like apples, peaches, or pears, spicy foods, and drinks containing artificial sweeteners. If a patient's stomach can handle some food, it's recommended to eat a mild, bland diet. A patient should try the BRAT diet, which stands for bananas, rice, applesauce, and toast. Even though the BRAT diet may not be better, more helpful compared to others, it can be less irritating to the digestive tract than consuming other foods. Another home remedy is using a heating pad, as it can ease abdominal cramping. But if it's uncomfortable for those who are experiencing vomiting and diarrhea, then it should not be used. There are over-the-counter therapies that can help relieve symptoms of Salmonella poisoning. Pain medications such as ibuprofen act as painkillers that help reduce body aches and pains. Anti-diarrheas like Imodium might decrease the abdominal discomfort caused by Salmonella, but it has drawbacks as it can extend the length of time one experiences diarrhea from Salmonella. Probiotics can treat symptoms of Salmonella. In 2013, microbiologists from the University of California found a probiotic strain used to treat irritable bowel symptoms that could comfort gut infections due to Salmonella. A strain of E. coli called Nissel 1917 could be helpful against the symptoms of Salmonella. If the Salmonella cases or causes are uncomplicated, then antibiotics are not recommended. While people usually recover from Salmonella within 4 to 7 days without medicine, the infection can persist. If that happens, then it's possible the patient has either the following: a compromised immune system, or the infection entered the bloodstream. In that case, healthcare providers may prescribe antibiotics such as amoxicillin, cefotaximine, and ciprofloxacin. But antibiotic treatments have risks, such as an increased chance of a relapse and extending the time period to the bacteria that lives in the human body. This can be as serious as the infected patient could also infect others who have Salmonella. Of course, there are ways to prevent Salmonella from spreading. When cooking, make sure you wash your hands, cutting boards, utensils, or anything that have been in contact with raw meat. Make sure you wash your hands in between handling different kinds of food. Speaking of which, make sure you wash fresh vegetables and fruit thoroughly before eating. And while cooking, make sure the cook temperatures are as follows: 145 degrees Fahrenheit for roast, 160 degrees Fahrenheit for ground meats, and 165 degrees Fahrenheit for all poultry. Make sure the refrigerator is below 40 degrees Fahrenheit. Keep hot foods hot and cold foods cold. Don't eat or drink anything that contains raw eggs or unpasteurized milk. And make sure you wash your hands with soap after handling snakes, lizards, or other reptiles. And that is it for this video presentation on Salmonella. If you have any questions, remarks, or anything, please post them on the discussion question 2 forum, in which I will be posting my video presentation on. I hope you have a good day, and take care of yourself.